Welcome back, everyone. Welcome back. Well, welcome back to the internet, I guess. Not really on campus, but hope you guys had a great holiday break. Got some rest. Hope you enjoyed yourself. Hope everyone was safe, of course. But the show must go on, and our show is almost coming to an end. So as you can see from the schedule, in front of us, we have 3 classes or 3 weeks left of material, so not a ton of time left. But there's, a decent amount of stuff to accomplish in that time. So we're going to be covering meiosis, sexual reproduction, and genetics today. So it's going to be a loaded class, but I think a class you guys will like. People generally like genetics. It's a pretty cool topic to discuss, the fun one to teach. So you guys will hopefully like that. In terms of what you got to turn in this week, there is peer review for your BioWiki. Everyone has to do this independently. I sent out an email. If you don't recall where the instructions are, I will show you. So, BioWiki materials. It's inside the project description file right here. It's one of the last things. So just in case you need a refresher where that is, it's due on Blackboard. Everyone's supposed to do this independently. And once I get all the peer reviews turned in, I will send out those peer review plus the review that I will do for you. So you'll get comments from me, you'll get comments from your classmates, and so on and so forth. So have lots of stuff to incorporate, and then your final draft is going to be due near the end of the semester. So that final draft, I expect you to incorporate all the comments, bare minimum that I've made, because I think my comments will probably be more substantive than everyone else's, because I'm the person that gives this assignment, so I know exactly what I'm looking for. So definitely want to make sure you incorporate all comments that you receive. And your classmates' feedback is also going to be valuable, because you have this person that is not your professor, right? So their knowledge bank is clearly smaller, and so if they don't understand something, it's going to be a good way to sort of make your wiki a little bit better. And then just we're going to cover respiration and energetics in the coming weeks. We have a few more activities left. In terms of lab work, we have this week, you have a lab exercise. It's going to be on meiosis, so it fits nicely with what we're going to cover today. And then the final 2 labs will be the virtual genetic lab. And you'll have 2 deliverables for me. You have to make a quick little PowerPoint presentation, basically showing me the results from your VGL1. And then you have a very, very brief lab report for VGL2 that is going to be due. But we will meet next Monday at 11. I already scheduled the time for it on Blackboard Collaborate. Just as a note, so I did schedule time. I originally was planning on doing this Monday, but I had a sick dog. He ate out of my compost pile, and of course, you get sick when you eat rotting garbage, basically. So, but next Monday at 11, definitely want to come, because I'm going to show you how to do all this VGL stuff. And if you don't come, you'll only get to see the recording. I won't, I'm going to show everyone how to use VGL and the way this works, but I'm not going to spend hours and hours if you don't come to class to show you how to use VGL. So make sure you come next Monday, or at the bare minimum, watch the video that I will post about this. And that's it. So almost to the end, not a whole lot left. You do have an exam, but not too much left. 1 homework, 1 exam. There's 1 extra credit discussion board. And that's sort of it. So we're almost there. We are almost there. Exciting stuff. And then, I'll see you guys next semester after that, because I'm assuming you're going to take biology 2 with me. So that's going to be fun. I'm excited for Bio 2. Bio 2 is much more up my alley in terms of materials, because we're covering ecology and all that fun stuff. So, but I digress. So if anybody has any questions, feel free to reach out to me via email. If you have any questions, you can drop them in the chat, or raise your hand, or speak out. But let's get started on meiosis. So before break, we talked about mitosis, right? So how we go from 1 cell to 2 cells. So today we're going to talk about almost the exact same process, but we're going to talk about it in the context of how we produce sexual cells. And when we say sexual cells, I don't mean the cells that make up your genitalia. I mean the cells that physically make offspring for the next generation. So we're talking about sperm and egg. So that's what we're going to be focusing on today, how meiosis works, how it's different from mitosis, and how sexual reproduction does work. So let's start off with the basics. Let's talk about sexual life cycles. So a sexual life cycle is made up of a combination of meiosis as well as fertilization. And so, sort of almost like the birds and the bees in a sense, but we're thinking about it from a biology context. So you start off with diploid cells. So these are your somatic or non-reproductive cells. Basically anything in your body that's not sperm or egg is a somatic cell. And somatic cells have two sets of chromosomes. They have two copies of chromosome 1, two copies of chromosome 2, and so on and so forth, thus making you diploid, right? So when we last time we were together, we talked about the idea of number of chromosomes being referred to N. Those that are haploid have N. Those that are diploid, i.e. two copies of every chromosome, are diploid. So just to bring that back to thinking about mitosis. Now, haploid cells are going to be our gametes, egg and sperm, and they only have one set of chromosomes. So just one copy of chromosome 1, one copy of chromosome 2, and so on and so forth. And the idea with this sexualized life cycle is your offspring inherit genetic material from two parents. So not like what bacteria do, right, where they have binary fission, asexual reproduction. We're going to be focusing on this idea of sexual reproduction. Now, we're focusing on this through the lens of mammals mostly, thinking mostly about humans. But I just figured I'd make mention there are other life cycles out there, not just living as a diploid organism and so on and so forth. When we talk about plants next semester, you'll see that many plants live as haploid organisms, unlike humans, which live as diploid organisms. And there's all sorts of weird sorts of sexual life cycles out there outside of this genetic information from two parents sort of scheme. So we're going to talk about what's sort of most important to you as an individual. But as I mentioned, diploid cells, i.e. having two copies of every chromosome, arise from the fertilization event where a haploid sperm fertilizes a haploid egg, producing a diploid zygote. The term zygote just simply refers to fertilized egg. And so that's sort of a blanket term. So for those of you that like fruit, a fruit like like an orange is just a zygote, right? It's just a fertilized egg, right? Most fruits are just fertilized eggs. That's sort of like one of the technical definitions of a zygote. But a zygote again is a fertilized egg, and in this case, we're talking about a diploid zygote. So we have two copies of each chromosome. One copy came from mama, and the other copy came from daddy. And this whole idea of fertilization brings forth this idea of sexual reproduction. And sexual reproduction is kind of an amazing thing from a biological standpoint. The vast reservoir of genetic diversity that humans have comes mostly from sexual reproduction. This process that we're going to discuss, as well as meiosis, again, a process we'll discuss today. And sexual reproduction is a really important way to introduce novel genetic material to a population. So bringing together two distantly related groups of individuals brings lots of diversity of genes together. But sexual reproduction, as we've sort of alluded to, is a process that involves meiosis and fertilization. And this is sort of that blanket term, right? You have to have meiosis first, and meiosis will lead to haploid zygotes, which in turn are fertilized, or one fertilizes the other, I should say, creating a diploid zygote. Now, there are, as I mentioned, some weird life cycles out there that don't have diploid phases or have very long haploid phases and so on and so forth. But we're thinking mostly about animals and mammals today. So in animals, the diploid state does dominate. And when we're thinking about sort of this process, we're thinking about a zygote has to be produced via meiosis. Fertilization has to occur. And once fertilization occurs, our friend mitosis takes over from there. But let's just look at this just very sort of simplified life cycle from a human perspective. have a male and a female. Biologically speaking, all of our somatic cells are diploid or 2N. Inside the ovaries and the testicles of men and women, you have the production of sperm and egg, which are haploid zygotes. And the sperm, as you guys probably all know at this point, fertilizes the egg, creating a fertilized egg, a zygote, which is now diploid. So going haploid to diploid. And then mitosis, as I've mentioned, takes over, taking that fertilized egg and turning it into the monster mass of cells that even a baby is, right? You know, babies are still made up of a few trillion cells. So lots and lots of mitosis to go from a single cell all over to a fully grown baby. It's actually would be a really fun exercise. Maybe the next time I teach this course, we'll do this exercise where you can calculate based upon your body weight how many mitosis events have occurred since your zygote stage, right? Because the bigger you are, the more massive you are, the more mitosis has to have occurred, right? You can't be 6 foot tall and have the same amount of mitosis events as someone who is 5 feet tall, right? But I digress. Okay, so to produce our zygotes, we need meiosis. And meiosis is very, very, very similar to mitosis. And we'll contrast those in a little bit. But meiosis does include 2 rounds of cellular division. We call them meiosis 1 and meiosis 2. Each of them has our prophase, metaphase, anaphase, and telophase stages, as well as cytokinesis. But there are a couple unique features to meiosis. And principally, we're thinking about synapsis, and you'll see what that looks like in just a moment. But this occurs during prophase 1, and we see homologous chromosomes become very, very closely associated to one another. And this causes the formation of what we call a synaptonemal complex. I hope I say that right. I never say things right, but and this formation of this complex is also called a tetrad or a bivalent. So what do I mean by that? Well, our chromosomes, we know we have that standard structure, right? That, you know, X-like shape that chromosomes all take. But we have, you know, the kinetochore, the centrosome, right? We have this whole thing, right? And they're bound, 2 sister chromatids are bound together, forming 1 chromosome. But what we're talking about with meiosis is we have this bivalent, and we have this synaptonemal, not gonna try to say it. We have this bivalent forming between 2 homologous chromosomes. So this would be like chromosome 1, and the bottom would also be chromosome 1. So we have this really intimate formation of this really intimate association of our paired chromosomes together during prophase of meiosis. And this bivalent complex is mostly just proteins. Just as a note, mostly just proteins. And so what does this look like? We have our chromosome, and there's a couple sorts of biology that we have here. So we have 2 lateral elements, which we call LEs. They're here in red, and they're connected to what we call a central filament, or CE. And these are connected by what we call transversive filaments, or TFs. And this is basically all just a giant pile of protein. Really all it is. As we sort of discussed early in the semester, proteins pretty much do all the things in your cell. So if you're going to form some structure, it's probably going to be protein. And so, but again, we are forming this bivalent complex. Molecularly, it looks a bit like this. And we have, it's basically all made out of protein, right? And it's just how we pair homologous chromosomes with one another. So that's an important feature of meiosis, but there are other things we're going to discuss. So while our homologs are paired during prophase 1, as we mentioned, genetic recombination, or this process of crossing over, does occur. Now, you probably have heard of crossing over before, but it's a really important aspect of genetic diversity. And I'll show you what it physically looks like in a second. But it's basically where you have 2 chromosomes exchanging bits and pieces with one another. The sites of crossing over, i.e. where 2 chromosomes swap genetic material, is called a chiasmata. Again, words. Chiasmata. So we have this, you know, sort of this terminology. And we call this first mitotic division, just as a note, we call it reductive division. And this does result in daughter cells that do contain 1 homolog from each chromosome pair. there is no replication of DNA between meiotic events. So meiosis 1 and meiosis 2, we don't have a complete replication of our genome because we're still going from basically 4N down to 2N, and then we're going to go down to N eventually. And then I'll just make mention that the second mitotic division does not further reduce the number of chromosomes, so it does separate the sister chromatids for each homolog. So let's dive into meiosis, and I'll show you crossing over in just a moment, but definitely have to sort of foreshadow it beforehand. Now, very much like mitosis, we have those standard sets: prophase, metaphase, anaphase, telophase, and eventually cytokinesis. But again, remember with meiosis, there is meiosis 1 and meiosis 2. Now, cells that are undergoing meiosis have a very similar interphase period to those that are undergoing mitosis. So they have the G1, S, and G2 phases. So we have, the replication of our chromosomes, the checks, right? We have all that fun stuff we discussed 2 weeks ago. But after interphase, so after G1, S, and G2, germline cells enter meiosis 1 as opposed to entering mitosis. So again, similar flow of information, but similar start, right? Interphase is the same between meiosis and mitosis, but meiosis is, again, a very different process than mitosis. So let's talk about these steps. Let's talk about prophase 1 of meiosis 1. So in prophase 1, the chromosomes physically coil tighter and become visible, the same way as it happens during prophase 1 in mitosis. The nuclear envelope starts to disappear, and then that spindle apparatus starts to form. Remember, each chromosome at this point is composed of 2 sister chromatids. And remember, during prophase 1, we have a synapsis, or that chiasma, forming. And remember, that formation of the synapsis, or the chiasma, is a process called crossing over. And this is genetic recombination. And that loaded term, right? Genetic recombination sounds kind of like a scary term. I don't know. It's a loaded word, right? But it's basically just the swapping of genetic information. That's all genetic recombination really is. And the homologs themselves physically exchange corresponding parts of their chromosomes with one another. So the alleles of a gene that were found on separate homologs can now be found on the same homolog. So that's sort of what we're going for, and I'll show you a picture of that in just one sec. But I just want to make note that this contact of this chiasmata is maintained up until anaphase 1. So this bridge is going to be basically occurring almost throughout the entire process of meiosis 1. But this is what we have. We have this crossing over event during prophase 1. We have essentially, remember, that formation of that bivalent bridge between our homologous chromosomes. During meiosis, metaphase 1, we have that crossing over event. So we have that chiasmata here, and we could see that they're physically just coming on top of one another. They're basically breaking their chromosomes and swapping bits and pieces. And you can see that's happening not just once here, but twice here, which leads us to having, again, we started with all blue chromosome. We ended up with a mixture of blue and red for this left chromosome. This right chromosome, we start with all red, and we end up with a mixture of blue and red. So we're just swapping genetic information between homologous chromosomes. Now, it might not seem like a kind of a big deal, but you are shuffling around alleles, because remember, chromosomes contain genes, right? And genes have different forms called alleles. And so by swapping alleles, you're seeing a shifting or a shuffling of genetic information. So you can kind of think of it like shuffling a deck of cards. Every time you shuffle a deck of cards, it's going to come out to a different order, right? Maybe the first card you pull is the ace of clubs, okay, and then you shuffle your deck again, and the next one you pull is the king of hearts, right? That's what really crossing over does. It essentially just shuffles the genetic deck. And again, that might not seem like a big deal, but it really is. So because of crossing over, and I'm going to throw some numbers at you because I think it's really astounding. Because of crossing over during meiosis 1, every time this occurs, you're almost guaranteed to produce a unique gamete. And the way this works is just by shuffling this around, right? So again, it's like shuffling a deck of cards. It's almost physically impossible every time you shuffle a deck of cards to get the same exact order. It's the same thing with crossing over. Every time it occurs during meiosis 1, you get a unique gamete. And so every time, basically the probability of getting the same gamete twice is basically a 1 in 1 trillion chance. So it does maybe that doesn't do it justice, the 1 in 1 trillion chance of getting the same gamete every time you produce a gamete, but it really is. And you think about this, like every sperm a male produces, every egg a female produces is likely a unique genetic variant. You are really 1 in 1 trillion chance to have happened, right? 1 in 1 trillion chance on your mom's side and 1 in 1 trillion chance on your dad's side. So you really are unique genetically speaking, which is kind of an amazing thing. Because you think about, like, for instance, like if I showed you a picture of my sister, you would like, really, you guys are brother and sister. And the reason we look so differently is because of crossing over. Same mom and same dad, same genetic material, but the shuffling of genes because of crossing over makes my sister and I look so different. And that's assuming that my parents didn't do something unethical with one another, but I don't know that. But I'm assuming that my sister and I are related with one another. So crossing over gives us this really unique set of genetic information. And it is really, really, really important to genetic diversity in humans and other large organisms. So I hope I've sort of hammered home the point of how important crossing over is during meiosis 1. It's a really important way to introduce novel genetic information. And when you take biology with me next semester, Bio 2, you'll see really how important novel genetic information is to an evolutionary standpoint. But now we're just thinking about the nitty gritty, the molecular biology behind all this fun stuff. But again, it is really important, genetically speaking, for pretty much everything that's big. It's not like bacteria where we mutate fast and can horizontally acquire genetic information. We rely pretty much solely on crossing over to give us novel genetic variation within our populations. But I'm going to stop talking about that because I could talk about that for a long time because I think it's a really fascinating part of biology. But let's move on from anaphase to metaphase. So the paired homologs during metaphase 1, they lock together following crossing over. So they basically lock together like this during metaphase 1. And the microtubules themselves from opposite poles, each physically attached to the homolog. So very much like metaphase 1 in mitosis, you have that locking on of microtubules, same process. And the important thing here is microtubules attach to each homolog, whereas opposed to in meiosis, they attach to each sister chromatid. So it's slightly different, same process, but slightly different in terms of how they attach. And you'll see how that comes to be in just a moment. The homologs, very much like in mitosis, are aligned at that metaphase plate. And so remember that metaphase plate is just an imaginary line right down the middle of the axis of the cell. So just you cut the cell in half, and that's the metaphase plate. And the orientation of each pair of homologs is on the spindle is random. And what do I mean by that? Well, they're randomly oriented. So you can have one gamete that's undergoing meiosis like this, another that looks like this, and so on and so forth. It's just random. So we'll talk about this in the second part of this lecture. We talk about independent assortment, but this gets at the idea of independent assortment of gametes. Alleles are independently and randomly assorted, and they're randomly assorted because of this random orientation during metaphase 1. All right, anaphase 1. The microtubules of the spindle shorten, so they start to contract and pull the chromosomes to the poles. That chiasmata, that bridge between our sister homologs, breaks, and the homologs are physically separated from one another, and they do start to move to opposite poles. The sister chromatids do remain attached at the centromeres, so they haven't detached yet. So again, very similar to mitosis, right? Nothing groundbreaking that we've discussed. But again, we're thinking about homologous chromosomes here as opposed to sister chromatids. Each pole ultimately gets a complete haploid set of chromosomes. And this is idea. We have this independent assortment of maternal and paternal chromosomes. They independently get sorted to one side or the other. So that's anaphase. Telophase, again, very similar to telophase in mitosis. The nuclear envelope starts to reform around each daughter nucleus. The homologous chromosomes finally start to separate from one another. The sister chromatids, remember, as opposed to mitosis where they're identical, the sister chromatids are no longer identical due to crossing over, right? That swapping of genetic material. And then cytokinesis may or may not occur right after telophase I. And eventually meiosis II does occur after an interval of variable length. And you'll notice that there's no interphase in between here, right? So we go right from the end of meiosis I. There's like a little bit of a break. Usually it varies. It can be, minutes to hours to days, depending on your species or what part of your life cycle you're in. But you don't have another interphase in between meiosis I and meiosis II. And meiosis II is essentially identical to meiosis I with a few little bit of caveats here. It does very, very heavily resemble mitosis as well. During prophase II, the nuclear envelope does dissolve and a new spindle apparatus forms. Metaphase II, the chromosomes lie on the metaphase plate. Anaphase II, the sister chromatids are separated from one another. And then in telophase II, the nuclear envelope reforms around 4 sets of daughter chromosomes. Cytokinesis then follows. And you notice there's no more crossing over here. There's no more chiasmata. There's no more anything, right? This is basically just mitosis, right? And very, 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 very straightforward. So nuclear envelope breaks down during prophase II. The spindle apparatus forms. Again, we have the lining of the metaphase plate. We have those sister chromatids are joined at the centromere, very much like mitosis. The kinetochore microtubules attach on to the center, the centromere. Anaphase, they're pulled apart. Telophase, the nuclear membrane starts to reform. But again, we're not at 2 cells now. We're at a grand total of 4. So we start out with 1 cell. It underwent meiosis I, didn't replicate its genome in between, went right into meiosis II, giving us 4 identical daughter cells. And we're not diploid cells here, right? There was no DNA replication in between meiosis I and meiosis II. So we're left with 4 haploid daughter cells. So 1 copy of chromosome 1, 2, 3, and so on and so forth. And so that's what we have. But the final result of meiosis is we have 4 cells, as I mentioned, each have a haploid set of chromosomes. In animals, these will develop directly into gametes. Now, this just looks like a cell, right? Sperm and egg are a bit different than your average everyday cell. So there is a level of development that has to occur after meiosis II concludes, at least in animals. Now, in plants, fungi, and many protists, those cells then begin to divide mitotically, producing a greater number of gametes and adults with varying numbers of chromosome sets. So clearly some funky stuff goes on here, but we're not going to really focus on that. We're going to keep talking about our animals in terms of what's most important for you guys to remember. Now, meiosis, very much like mitosis, is error prone. They're not as common as you would think, but they certainly occur during meiosis. And so one of the big errors during meiosis is non-disjunction, and it produces a lot of really common genetic disorders, such as Down syndrome. But non-disjunction simply refers to the failure of homologous chromosomes to move to opposite poles during either meiotic division. So it gives you 1 or 2 extra copies of a given chromosome. So you could have 3 copies of chromosome number 21, or 4 copies of chromosome number 24. Or I should say your sex chromosomes, right? So that's the idea. Adding non-destruction leads to extra or too few chromosomes in one of your gametes. This gives us a technical term, which is called aneuploid gametes, and these are gametes with missing or extra chromosome. And just as a note, the most cause of like miscarriages and what we call spontaneous abortions in humans is due to aneuploid gametes. So gametes that just don't have quite the right number of chromosomes. Now, there are some types of gametes that have extra numbers of chromosomes that are tolerated. So I mentioned Down syndrome. Down syndrome has 1 extra copy of the 21st chromosome, and those produce viable offspring. So not all aneuploid gametes are deadly, but some, and because some clearly can produce viable offspring. So, All right, so let's contrast meiosis and mitosis for this sort of heading towards the end of this first part of the lecture. So meiosis is physically characterized by 4 distinct features versus mitosis. So the homologous chromosomes are paired and they cross over during meiosis. That doesn't occur during mitosis. Remember, this first example only occurs during meiosis 1. The second thing is that sister chromatids do remain joined at their centromeres and segregate together during Anaphase 1. Again, that does not happen in mitosis. The kinetochores of sister chromatids attach to the same pole in meiosis 1. Doesn't happen in mitosis. And DNA replication is suppressed between meiosis 1 and meiosis 2. Because remember, if we replicated our DNA, it would just produce a 2N organism, right? A diploid organism. So we need to keep DNA replication suppressed between meiosis 1 and meiosis 2 so we can produce haploid gametes, because we need haploid gametes, right? We can't have diploid gametes. That's not going to do us any good. So, but let's look at this alignment of chromosomes between meiosis 1 and mitosis. So remember, during both processes, the homologous chromosomes, or the sister chromatids, physically pair up along the metaphase plate. In meiosis 1, we have a pairing of homologous chromosomes, i.e. 2 copies. So this is 2 copies of chromosome 1. This is 2 copies of chromosome 1. Right? That's what we're thinking about during meiosis 1. Mitosis, we just have 1 copy of chromosome 1 pairing, 1 copy of chromosome 1 pairing with one another. And you can imagine the other copy of chromosome 1 would be here and here. So very, very different alignment of chromosome. Here we have homologous chromosomes. This one we have sister chromatids. So let's look at this sort of this whole piecemeal. Again, we have meiosis on the top, mitosis on the bottom. We have a parent cell. It's 2N. 2N. We have. Oh, this image got a little funky here. That's okay. We have a 2N, and remember, before meiosis 1 or mitosis can start, we have to have the duplication of our genome here, and they're very, very similar in terms of what they do. Right? We have metaphase, so on and so forth. But remember, the key difference between the two here is the alignment of these chromosomes, the crossing over event during meiosis. And remember, mitosis only produces 2 identical daughter cells, whereas meiosis does produce 2 identical daughter cells after telophase, but we skip replication of the genome and head right into meiosis 2, where we produce where we essentially have 2 rounds of meiosis again. There's no crossing over, but we have, very much like mitosis, the alignment of the sister chromatids along that metaphase plate, separation during anaphase, and then after telophase and cytokinesis, we produce 4 daughter cells. They are not identical at this point. I'll make mention that these 2 daughter cells here are not identical either. You can see their genetic information does vary quite a bit. But we do have 4 unique daughter cells, each with half the amount of DNA. So 1 copy of chromosome 1, 2, so on and so forth. So very similar, right? The terminology is very similar, but the key is what is different between them. And so it's easy enough to remember the steps, like, oh, an anaphase, you're starting to get pulled to the poles, but it's sort of remembering what's happening to those chromosomes in meiosis, which is really important to sort of distinguishing between meiosis and mitosis themselves. So as I've mentioned, homologous pairing is specific to meiosis. And the sort of interesting thing about this is, and despite the fact we've been studying this for decades, how homologous chromosomes find one another and become aligned is actually not understood. We don't understand how these chromosomes find one another during meiosis. We always sort of make the assumption that it's the things we learn in these introductory classes that we know, like everything about it. But the reality is, like, so much of this basic biology is still kind of a mystery. But sort of an active area of research is sort of this idea: how do homologous chromosomes find one another? And the most recent research about this seems to be like a specific class of proteins help them find one another. But again, we don't really quite know how homologous chromosomes find one another. Now, sister chromatid cohesion is similar to that of mitosis, but it does involve meiosis-specific cohesin proteins. So you'll remember that the attachment of homologous chromosomes to one another involves a set of proteins called cohesins. And so you might remember we talked about SCC1 protein in mitosis. Well, we know that in meiosis, the protein REC8 does replace it as part of that cohesion complex. So similar structure of the protein, similar function, different genes that encode them. So just making mention of that. And those synaptonemal complex proteins are identified in a diverse number of species, but the proteins actually show very, very little sequence conservation. And what does that mean? It just means that my synaptonemal proteins are very, very different than, say, my dog's synaptonemal proteins. Whew. That was a tough one to say. All right, so sort of heading toward the ends of this part of the lecture, the centromeres of our sister chromatids remain connected. And so the maintenance of sister chromatid cohesions as a centromere during all of meiosis 1, and we do see a loss of cohesion from the centromere arms during Anaphase 1. We have actually this kind of interesting protein. They're called chigoshin. It protects cohesin, I should say, from separase mediated cleavage during meiosis 1. A lot of words saying we have a special protein called chigoshins that protect essentially from the separation of these chromosomes from one another. Because remember, during mitosis, the sister chromatids are physically yanked apart. But in meiosis, you have to keep them together, and then they're held together again by those cohesions, but the cohesions are protected by our chigoshin proteins. Ugh. Word. Words are difficult today. And so sort of one of our last things we'll discuss in this part of the lecture is the sperm versus the egg. So how do we go under this process? How is it different between sperm and egg? And the reality is it's not that different. It really isn't. The meiosis 1 and meiosis 2, they're identical between sperm and egg. So that's a good thing. But this whole process of producing gametes is called gametogenesis. That's just a technical term. But to produce an egg, the gametes undergo oogenesis, oo meaning like oocyte, as in egg, and then spermatogenesis, sperm meaning sperm. So you have a very similar process. You have the primary oocyte or the primary spermatocyte. That's how you start. They undergo meiosis, not just once, but twice. And the key distinction here between the spermatogenesis and the oogenesis is that spermatogenesis, you have two sort of key things. You have the production of 4 gametes from every round of meiosis 1 and meiosis 2. So that's an important thing. For every primary spermatocyte, it will produce 4 sperm cells. Now, the other big difference is you'll notice that the sperm have tails, right? And so I'm sure you guys, I don't need to tell you the birds and the bees sort of situation, but the sperm has to swim to the egg to fertilize the egg, right? And so that's sort of a key difference, right? You can't really swim in a cellular way without some sort of tail-like structure to propel you. Think of it like a fin, like a tail of like a whale or something, propelling them through the water. So sperm do need that. So there is this level of maturation that occurs to produce the tail for the sperm, or I should say the technical term would be the flagella of the sperm, not the tail of the sperm. So that's the sperm. Now, the other big difference is that oogenesis, so the process of producing eggs, produces only one mature ovum or one mature egg per every primary oocyte. So whereas sperm produce 4 sperm every time, eggs only produce 1. So they produce 1 mature egg that's much larger than all the other what we call polar bodies, which are basically like very, very tiny cells. They have all the correct genetic information, but they're not functional cells. They eventually just die. So every time a biological female produces 1 mature egg, there are 3 polar bodies that are basically like dummies. They don't do anything. So big differences here, right? The mature ovum is just like a regular cell, but we have sperm that again are very much, you can think of it like tadpoles, right? Very, very, distinct in terms of their biology versus an egg. So, so that's a sperm and an egg. And the other last thing, just to make mention of, is that meiosis does produce cells that are not genetically identical. So we talked about crossing over introduces tremendous amounts of genetic diversity. Remember that there's a random orientation of chromosomes during meiosis 1. Because of those two things, meiosis rarely, if ever, produces cells that are identical. Remember, every egg is a 1 in 1 trillion chance of being a similar egg. Every sperm is a 1 in 1 trillion chance of being identical to another sperm that's produced. So basically, every sperm and egg that anybody has ever produced is likely unique, which is really cool to think about. Right? You really are, like, genetically speaking, you are kind of one in a trillion, right? You're better than 1 in a million. You're 1 in a trillion. So if you ever wanted to feel special, it's definitely a way to, to make yourself feel a little bit special, because you are a genetic abnormality relative to the rest of the people out there. And I say that as a good thing. Being a genetic abnormality is a good thing. But as we'll discuss next semester, this results in lots of unique genetic variability, lots of unique traits, and this is essential for evolution. So we'll talk about the implications for meiosis next semester in biology 2. So I'm sure I'll see you guys all there. I think everyone has signed up at this point. But if you haven't signed up for classes, definitely sign up for classes. So that's part 1, meiosis. Now the pairing of meiosis really kicks off when we start to think about how we inherit genetic information. What are the rules that dictate how traits are passed on? What are the rules by which you are you? What are the rules by which fertilization works? So that's really the second part of today's class, and which is why I like to pair these two things with one another, because they really feed off of one another. So we're going to be spending the remainder of our time talking about genetic inheritance and what we call Mendelian genetics. So this second term will become much more clear by the end of the class, or maybe it's clear right now. You might already know who Mendel is, but if you don't know who Gregor Mendel is, you will by the end of today's class. And I like to start off, it depends on what I'm teaching, but I like to start off with a picture like this. We're all the same genus species, we're Homo sapiens, and there is a huge amount of genetic variability within the human population in terms of physical traits. Normally, when I teach this class in person, I ask everyone, oh, look around the room. Do you see really anyone that looks quite like you? And the answer to that is usually not really, right? No one really shares meaningful amounts of characteristics with one another. And how you get your traits is clearly derived from your DNA, but how did you get those traits? Well, clearly it has to do with fertilization, right, and meiosis, but how does inheritance work? How do you look more similar to your parents, presumably, than you do to your neighbor? How do you look more similar to your sister than you do to, say, your cousin? And so this is how we get at this idea of answering those questions, is via inheritance and genetic, Mendelian genetics. Now, there's a really interesting history behind heredity. Uh, and it's led to a lot of really stupid things over time. I won't talk to you about those stupid things, but you definitely should look up, these arcane ideas of like the more arcane ideas about how inheritance worked in humans. They were really, really stupid. Like they were really stupid. So definitely look that up. We're not going to talk about that today, but definitely look it up. It's a funny read. But prior to the 20th century, there were two sort of primary concepts that were the basis in the scientific community about heredity. Now, heredity itself occurs within species. That makes sense. You can't inherit genetic material from your cat. Doesn't work that way. And traits are transmitted directly from parent to offspring. So pretty straightforward stuff, right? You get your traits from parents, and you can't get your traits from your cat. That's pretty straightforward. That's clearly not all there is to heredity, but that's sort of the these big, widespread ideas, scientifically speaking, prior to the 20th century. Now, historically speaking, people thought traits were born through fluid and they were blended in offspring, which is kind of a weird thing. Um, so like you think about like just blending of biological fluids together to produce an individual. That's the idea, which is why his pre 20th century ideas about heredity are really stupid, at least from our point of view, because we know so much. But I digress. Now, this does bring about this idea of a paradox. So if blending does occur, why don't all individuals look alike, right? So old school ideas of heredity don't work, but this idea that heredity occurs within species, that traits are transmitted directly from parent to offspring is 100% true, right? You only get your genetic material from your biological parents. Now, in terms of understanding heredity, we often turn to plants to understand how this works. So we're going to talk about a lot about plants today, just because plants are really easy to work with. The genetic systems are easier. They have life cycles much faster than humans. So plants are, they're really good model organisms to understand genetics. So if you guys ever take genetics with me here at Fisher, you'll get to play with some plants in genetics. So just as an FYI. Now, back in the 1760s, we had Josef Kohlreuter, I believe that's how you say his name. He was playing around with tobacco plants, and he was producing hybrids of tobacco plants. And what Kohlreuter was seeing, he was seeing differing plants that were different from their parents, and he was seeing this huge level of additional variation observed in what we call the second generation of offspring, which contradict the idea of direct transmission, right? Like directly getting your traits from your mom and your dad. And so just as a note as to what second generation means, just because we're going to see it again. So, my parents, they produced me. So I'm the first generation from my parents. And if my wife and I have a kid, that kid will be the second generation from my parents. So just as a note as what I refer to as second generation or first generation. It's all relative to the system you're talking about, right? If I have two plants and they make offspring, that's generation one. And if I force the offspring to have offspring, that's generation two. So now, fast forwarded, 63 years, we had T.A. Knight. He was a scientist. He was working with the garden pea, Piscium sativa. Um, you know them, pretty closely as like snow peas. They're delicious. They really are. It's the only type of peas I actually like. Well, that's actually not true. I'll eat all peas, but these ones I will readily seek out to eat because they're so tasty. Now, what T.A. Knight did is he crossed two pure breeding strains. Now, what do I mean by pure breeding? Now, this is a term we're going to see a lot going forward. Now, true breeding means it has two alleles. So remember, these are very much like us. They have two chromosome 1s and two chromosomes 2s. Remember, chromosomes that are identical, right? Chromosome 1 and chromosome 1. They each have the same exact genetic information. Any variability in the genes between chromosome 1 and chromosome 2, or even if it's similar variability, we call those alleles, right? So each copy of a given gene is called an allele. Now, when you say something is pure breeding, you have two chromosomes. They have identical copies of the same allele. So in this case, you could pretend you have an allele that encodes for green color. You could pretend that we have that same green color allele on one chromosome and the exact same green color allele on the other chromosome. So just as a note. Now, what he did, he had two pure breeding. Remember, two identical copies of the alleles on the chromosomes. His first generation, he would cross them. So you have, say, like one that was yellow and one that was green. He would cross them together. And his first generation, he would see that they resembled only one parental strain. So if we pretend he crossed yellow and green together, he would only see green out the back end. However, in the second generation, where we cross two individuals from the first generation, he would see a mixture of green and yellow peas. So now, I think it's before we go and talk about Gregor Mendel, I think it's important to just sort of put this idea into context, right? We're talking about this knowing about DNA. We're talking about this knowing about alleles, right? These guys had no idea what that was, right? Remember, we didn't know the structure of DNA till the 1950s. Till Francis and Franklin and Crick and Chargaff and all those guys, right? We didn't know any of that. So these guys were talking about ideas without any real context for what was transmitting this traits from generation to generation. We know it's DNA now, but they didn't know any of that back then, right? So this system makes a lot of sense to us now, but remember, back in the 1820s, they don't know. So just wanted to put that in context, because I think it's important to interpret history with context, especially from biology standpoint, simply because we just know so much more, right? And we know so much more because these guys had no idea what they were doing, and they were doing all this crazy, unique science back in the 1700s and 1800s. Now, the star of our show today is going to be our friend Gregor Mendel. Now, he was an Austrian monk, handsome kind of dude, I guess. I don't know. He doesn't have a mustache. I don't know. I don't know if I can trust a. I don't know if I can trust a pre-1900s 1900s scientist if he doesn't have a sweet mustache like all the other scientists we've looked at. But that's Gregor Mendel. Now, Gregor Mendel, very much like T.A. Knight, as we discussed in the previous slide, he worked with peas. Peas. So nothing exciting, not working with chimpanzees or anything like that, but he worked with peas. Now, peas still are a model organism, and that's an important sort of term. We've sort of defined model organism. Oops, sorry about that. Prior to today's class, but model organisms are organisms that are really easy to work with. Those that are genetically tractable, they have short lifespans, things like that. So Mendel worked with his model organism, which was peas. But Mendel more specifically worked with peas because other research showed that you could produce hybrids of peas. There was many pea varieties were available at the time, and obviously still are. The peas were small and easy to grow, and the peas can self-fertilize or be cross-fertilized. And what does that mean? Well, self-fertilization would be taking the pea's own sperm and fertilizing the pea's egg within one plant. Whereas cross-fertilization is if I take pea plant 1, I take its sperm and I fertilize the eggs of pea plant number 2. So pea plants can go either way in that respect. So just an important thing, right? You can self-fertilize or you can cross-fertilize with our peas. And that's an important, and you'll see why that's important in just a moment. So this is Gregor Mendel. As I said, he's going to be the star of our show going forward. Now, Gregor's experimental methods were pretty straightforward, which is good for basic biology courses such as these. Now, Mendel's had 3 distinct stages during his experiments. The first, he would produce true breeding or pure breeding strains for each trait he was studying. Remember, true breeding or pure breeding just means that when we're looking at the alleles, it has 2 identical alleles for that given trait. So 2 alleles for green peas or 2 alleles for yellow peas. That's what Mendel was getting. He didn't know that, but conceptually that's what he was going for. Now, what he would do, he would cross-fertilize true breeding strains having some alternative forms of a trait. So green with green, instead of green with green, he would cross green with yellow, or wrinkled peas with smooth peas. And he would do reciprocal crosses as well. So if he took, the sperm from a green pea and fertilized the sperm of an egg of a yellow pea, he would then take the sperm of a yellow pea and fertilize the egg of a green pea. So he would do this reciprocally, just to make note of that, because we'll talk about why that matters later on. And from there, he would allow the hybrid offspring to then self-fertilize for several generations, i.e. one plant sperm fertilizes its own egg. And what he would do is he would count the number of offspring showing each form of the trait. So every time he would produce a set of offspring, he would count numbers. And so I told you this part is genetics, and a huge part of genetics is simply counting. It really is. Knowing what the distribution of traits is in your new population, i.e. how many green plants do I have and how many yellow plants do I have, is basically the foundation of genetics. How does frequencies change from generation to generation? That's what's really important for genetics. So let's look at this. Let's look at some pea plant biology. We'll look at this again next semester when you take Bio 2. We're gonna have basically like 5 hours of plant biology at some point. So guys are gonna love that. But let's just look at the biology of his pea plants. They have the petals like all angiosperms do, so flowering plants. And inside the flowers, remember flowers are used for reproduction, right? We use them to look pretty, but flowers are really just used for reproduction. Now, Inside, we have the carpel, which is comprised of the stigma, which is the sort of the end cap here, and the style, which is basically the canal, almost you could sort of call it. So that's our female parts, and then we have the anthers, which are so these sort of like lollipop sort of looking structures. And so we have male and female. And now, very much like us, there's egg inside the carpel and sperm inside the anther. Now, in plants, sperm is called pollen, just as a note. So for those of you that are like me and you get really bad allergies in the spring and the fall, you're allergic to plant sperm. So that's fun. It's a terrible thing to think about, but that's what it is. So this is our basic plant biology. Anthers, carpel, remember it definitely for next semester. It's not really that important right now, but definitely important to understand the biology that we're working with. Now, what Mendel would do is he would cut the anthers away, because if you left the anthers like this, they could potentially fertilize the carpel. So he cut them away, and then he would use the pollen obtained from a different color flower to pollinate his original flower. And then the progeny themselves would result in purple flowers in this case. So he took a purple flower, crossed it, so this would be a female purple flower, crossed it with a male white flower, again, just by using a paintbrush to transfer the pollen from one plant to the other, and producing offspring. So in this case, purple with white gives you all purple at the back end. So this is the system. Now, if he was going to self-fertilize a plant, he would simply take his paint. He would still cut the anthers away, but he would take his paintbrush, take the pollen from the anthers that he cut away, and fertilize the style and the stigma of the sort of the original plant. So, same process in terms of self and cross fertilization, but conceptually they vary slightly. So, this is what Mendel did, and Mendel did this with thousands of plants. So you have to imagine Mendel was a pretty meticulous and pretty, I'm going to go with crazy kind of guy, because doing thousands of these crosses where you're using a paintbrush to transfer pollen must have been maddening. But he did it, and clearly we're talking about it, so clearly it's important, and it definitely is. So let's get into some of these crosses, right? So the first cross we're going to talk about is what we call a monohybrid cross. Now, a monohybrid cross is where we use a cross to study the variability in only two variations of a single trait. So if we have, as in this last slide, this is a monohybrid cross. We cross purple with white. So we're just looking at one specific set of alleles that control one specific trait. Now, this specific trait in this case is going to be flower color. Fun stuff. So that's what a monohybrid cross. Mono meaning one, and that one refers to a single trait. Now we'll talk about a dihybrid cross later, but we're looking just right now as a monohybrid cross because we're looking at specifically just one trait. And you can do this in plants because typically speaking, for the traits that Mendel looked at, one gene or one set of alleles corresponded to one trait. And there's no weird linkages going on here. So he started off with lots of monohybrid crosses. Now, what Mendel did, he produced true breeding or pure breeding pea strains for 7 different traits. He looked at flower color, seed texture, pod color, pod shape, flower position, plant height. So he looked at those 7 traits. Now, simple plants, right? Peas are pretty simple, right? This is simple, right? But the variability is a lot, right? There's a lot of genetic variation in this one species. And the important thing for Mendel is that each trait only has 2 variants. So for flower color, he would only have purple and white. For seed color, he'd only have green and yellow. For texture, he would have smooth or wrinkled, and so on and so forth. So many traits, but only 2 types of traits. Now, what he just to sort of show you these physically, again, we have flower color, seed color, texture, color, so on and so forth, right? So this is what he had. Each had 2 distinct traits. Now, just as a reminder, we call these distinct physical traits, we call these the phenotype of an individual. So a phenotype is purple. The phenotype is white. Now, each phenotype has an underlying what we call genotype, which is just the genetic information, right? What allele does it have? So this is what Mendel had, 7 traits, each with 2 specific variabilities on it. And kind of a nice system, right? Easily identifiable traits, easy plants to work with, tractable genetically. And the cool thing about this is you don't need to understand the underlying genetics to really understand Mendel's system. You can just physically see, right? You can physically just see. And the one thing I'd like to notice is, like you all to notice, is that you see these ratios. So 3.15 to 1, 3 to 1, 2.96 to 1, and so on and so forth. And so what he's actually, these ratios are the ratios in the second generation. So he took what this system, just as a reminder, he took 2 pure breeding plants, so 2 pure breeding white, and crossed it with a pure breeding purple. And the resulting offspring would be the F1 generation. Now, if you took 2 organisms from that F1 generation, or that first generation, and you crossed them together, you would get the F2 generation. That's what this sort of third column gets at. And you'll notice, and this will become apparent in a few moments, the ratio is always about 3 to 1. And we'll get into why that ratio is important in just a moment in the F2 generation. Now, I mentioned this idea of this F1 generation. The F stands for filial. So we call F1 generation the first filial generation. And this is the offspring produced by crossing 2 pure breeding or 2 true breeding strains. So for every trait Mendel studied, all F1 plants only resembled one parent. So we get some important terminology here. He referred to this trait as the dominant trait, and the alternative trait would be the recessive trait. And then the final important thing he saw was there was no plants that had characteristics that are an intermediate between the 2 parents. And what do I mean by that? Well, if I cross purple with white together, if you took a purple marker and you colored over it with a white marker, you'd have like a reddish color, right? He saw no like intermediate colors here, right? He saw no intermediate seed colors or pod colors or heights or anything like that. He would just see 2 distinct phenotypes. Now, as I've mentioned before, the F2 generation, or the second filial generation, he took the offspring resulting from the self-fertilization of F1 plants. So again, purple flower, purple with a white flower, he crossed them, producing the F1 generation. He would take a F1 generation plant and self-cross it with itself, so its own sperm into its own egg, producing a F2 generation. Now, the cool thing about the F2 generation is that in the F2 generation, it's a bit different, right? And although the traits were hidden in the F1 generation, the recessive trait, i.e. what you weren't seeing in the F1 generation, had reappeared among some individuals in the F2 generation. And he counted those proportions. So he would look at the ratio of purple to white, or yellow to green, and so on and so forth. And he always found about a 3 to 1 ratio. And what does that mean? Again, for every 3 purple flowers he would find, i.e. this would be the dominant trait, he would find 1 recessive white flower. Only in the second generation. Now in the F1 generation, he would only find the dominant trait. He would never see the recessive trait. Because, as we'll discuss, the genetics of the dominant trait overrides the genetics of the recessive trait. Now, so when we looked at the physical genotypes, i.e. the genetic information, that 3 to 1 ratio is actually a 1 to 2 to 1 ratio. And I'll get into that in just one second. But so when we looked at the F2 plants, 3/4 of the plants were the dominant form, so i.e. purple flowers, and 1/4 of the plants were the recessive form, i.e. white flowers. So the dominant to recessive ratio was 3 to 1. And what we're looking at here, remember, is only phenotypes, right? So the phenotypic ratio was 3 to 1. However, the ratio is actually a bit different. It's actually not 3 to 1. So the phenotype is 3 to 1, but the genotype is actually 1 to 2 to 1. And what do I mean by that? Well, one plant for every 4 plants you would produce, one plant would be true breeding dominant plant, 2 plants would be a not true breeding dominant plant, and 1 plant would be a true breeding recessive plant. So let's just conceptualize this. Conceptualize this experiment. What are we actually looking at? So we have the parent generation, a true breeding purple plant and a true breeding white plant. So they have 2 identical copies of the allele that controls flower color. What he would do is cross-fertilize them, so mate white with purple. And all the offspring in the F1 generation would appear to be purple. But remember, these offspring are not pure breeding or true breeding like the parent. They're a mixture of purple and white. Because remember, all offspring are just a mixture of their 2 parents. That's cool. Now, he took those F1 generations and he self-crossed them, i.e. he took the sperm of one plant and crossed it with the egg of the exact same plant. And what he would get out the back one is that 3-to-1 phenotypic ratio, where we had 3 plants that displayed the purple or dominant phenotype, and 1 plant that would show the white or recessive phenotype. So that's our F2 generation. But the important thing here is if he crossed them again, he would actually get in the F3 generation a very different genetic ratio. He would get a 1-to-2-to-1 genotypic ratio. And again, that means one of these plants is going to be dominant, i.e. pure breeding purple. One of these plants is going to be recessive, pure breeding white. And 2 are going to be non-true breeding and what we will call heterozygous plants, i.e. they have a mixture of alleles from white and purple. They don't just have alleles for purple or alleles for white. They're a mixture. They're heterozygous for alleles. So what can we conclude from Mendel? Now, his plants showed no intermediate traits. That's important. Each trait is intact. It's discrete, meaning there's no mixing going on. That's an important part of Mendel's. If his colors mixed together, Mendel's experiments would have failed, but his traits were distinct from one another. Now, for all 7 traits Mendel studied, each pair, one trait was dominant and the other was recessive, meaning that if you had the allele for purple and the allele for white in the same genome of one plant, the purple allele would override the white allele, basically masking the presence of the white allele. That's important. Now, remember so in that F2 generation, remember we saw that 3-to-1 ratio of phenotypes, but remember the underlying genotype was 1-to-2-to-1. So let's get to Mendel's 5-element model. Now, first is the paraments transmit discrete factors. That's what he called them. We call them genes, our DNA. He did not know what a gene was at the time, but he called them discrete factors. We now know that they're genes. So the parents transmit genes from themselves to their offspring. Each individual receives 1 copy of a gene from each parent. So that's sexual reproduction, right? Now, not all copies of a gene are identical. So remember, the variant on a gene is we call an allele. So alternate form of a gene is called an allele. When an individual is homozygous, they have 2 of the same allele. When an individual is heterozygous, they have 2 different alleles. Now, when we talked about these true breeding plants, our true breeding plants would have been homozygous because they had 2 copies of the same allele. So 2 purple alleles or 2 white alleles. Our heterozygous, i.e. that F1 generation, would have 1 copy of the purple gene, 1 copy of the purple allele and 1 copy of the white allele. So just putting what we talked about previously into context. Now, the other part, and we discussed this already, but I'll just reiterate it, the alleles themselves remain discrete. There's no blending of traits, right? We didn't have purple and white mixing together to give us red. We did not see that. And then finally, we have the presence of alleles does not guarantee expression. Now, What Mendel saw is that there was one allele that was dominant over the other. And so what this means conceptually is if I have the purple allele, which is dominant to the white allele, present in the same organism, the dominant allele, i.e. the purple one, will always be expressed, while the recessive one, i.e. the white allele, will always be hidden by the dominant allele. And just as a note, in terms of how your genome works, you only express one copy of each allele. You typically don't express both copies of your alleles. There's very few traits that you actually physically express both. So you always only express, and if there is dominant one and a recessive one, you will always express the dominant one. So, and the two sort of final sort of terminology that we've already discussed. We have the genotype, which is the total set of alleles inside of a single individual. This is what we call our genotype. And then the phenotype, as we've discussed, is that physical appearance. Purple, white, round, smooth, so on and so forth. So that's Mendel's model. Simple, but really, really fundamental to understanding how inheritance works. Some genes are dominant over others, but they're passed down from parents to offspring. It is inherited, right? And Mendel's model was great. He had discrete traits. There was no mixing here. Now, we have to next talk about another important contribution of Mendel, which we call the principle of segregation. Now, this gets at this idea that we have two alleles for a gene segregate during gamete formation and are then rejoined at random from one parent during fertilization. So if you just sort of conceptualize what happened in meiosis, right? We started off with a diploid organism, right? Makes copy of the genome, produces two identical or almost identical daughter cells, and then those go on to produce 4 gametes, or 1 if you're talking about an egg, right? So that's where we started, right? Now, during meiosis, that segregation, again, happens randomly, right? So each resulting zygote gets a different copy, or a slightly different copy of each chromosome. So that each allele does segregate independently. But remember, every time you have a fertilization event occur, you have chromosome 1 for mom, chromosome 1 for dad. They come together, producing the offspring in fertilization. And just as a make one more mention of this, Mendel had no idea about chromosomes or meiosis. That hadn't been described yet. So he was describing this principle based physically on what he could see in his P's phenotypes. That's all. So that's Mendel's big contributions. Now, we're going to talk about a really valuable tool for you to understand these crosses, and this is what we call a Punnett square. It's one of my favorite things to do. I really enjoy playing around with Punnett squares. They are fun. Now, pretty cool. Now, Punnett square simply refers to looking at what happens when you cross two individuals with one another. So in this case, we have a male and a female. You can see both the male and the female, they have 2 alleles. So they have one allele that we've deemed big P and another allele that we've termed little p. So generally speaking, when we make an allele big, i.e. capitalize it, it's going to be the dominant allele. And when we make an allele little, it's going to be the recessive allele. Now, based upon what I just told you, based on this idea of segregation, each gamete will only inherit one allele from its parent cell. So when a father produces gametes, and we have just one, we're thinking just about one gene here with 2 alleles, each gamete has the 50% probability to get a big P and a 50% probability to get a little p. So, and that's the same for the mother and the father. And so if fertilization was going to occur between these two, it would be either you get a big P from your mom and a big P from your dad, or you get a little p from your mom and a big P for your dad, and so on and so forth. So we draw a Punnett square, just a 6 celled box here. where we put one organism on the top. So in this case, we have the male on the top, so it would be the columns, and the female would be on the rows here. You could switch it up. It doesn't really matter. You could have females on top, males on the bottom. It doesn't matter. But the idea is you populate this box by saying, okay, in this corner, we'll start here where it actually already done it for us. In this bottom right here corner, we get the little p allele from mom and from dad at the same time. Thus, any offspring that gets this will be little p, so having two copies of the recessive allele. Same thing in this box, little p from dad, big P from mom, big P, little p. So it's a heterozygote. This bottom box here, little p from mom, big P from dad, cool beans. And then finally together, we have big P plus little p, giving us big P, big P. So this is how we use a Punnett square. Very, very straightforward, but a very valuable tool for you as an individual to understand how to sort of think about what the sort of the distribution of phenotypes and genotypes is potentially in a cross between two individuals. Excuse me. So let's apply Punnett square to analyze Gregor Mendel's crosses. So remember, he took two true breeding parents, right? So in this case, we're thinking about flower color. So he took a pure purple flower and a pure white flower. Now remember, purple is dominant over white. So in this case of purple flower, we'd have a homozygous dominant big P, little p genotype. For our white parent, we'd have homozygous recessive little p, little p. So this is what our cross looks like. Big little p multiplied or crossed with big P, little p. And so we just fill out the Punnett square. The purple parent only can give big P's and the white parent can only give little p's. So all the existing crosses, all the existing offspring can only have big P, little p. Because remember, that's all the parents could physically give. That's it. And so you can see the resulting genotypes. We have 4 individuals. They're all purple, but they all are big P, little p. It's pretty straightforward, right? So that's exactly what Mendel saw, right? His F1 generation, all purple. And you can see from our Punnett square, they're all heterozygous. Remember, heterozygous just means I have one copy of an allele that's different from the other copy of the other allele. So big P and little p. That's straightforward. Now, remember, Mendel also crossed 2 of these heterozygotes together. So we have big P, little p, crossed with big P, little p again. Now, remember what Mendel saw in that F2 generation. He saw a 3-to-1 ratio. That 3-to-1 ratio is 3 purple flowers to 1 white flower. That's the phenotypic ratio. But when we look at the genotypic ratio, that 1 to 2 to 1, you see we have 1 big P, 1 little p, and 2 heterozygotes, big P, little p. And so, again, our Punnett square, simple way to understand what happens when you cross these 2 specific genotypes with one another. Really straightforward, but a really fun. Well, I think it's fun. I love doing Punnett squares. They're really enjoyable to me. But a really simple way to understand what happens when you cross 2 genetically identical or 2 genetically different individuals with one another, so you can get both genotypic and phenotypic ratios simultaneously. So what we've been talking about thus far is what we call dominant recessive inheritance. So some human traits are controlled by a single gene. Not a super common thing in humans, but there are many traits that are controlled by a single gene. So something like eye color, controlled by multiple genes. Something like hair color, again, controlled by multiple genes. But there are some traits within humans that are controlled by a single gene that has multiple alleles. Now, some of our genetic traits, again, exhibit dominant and recessive inheritance. And so we can actually physically do a slightly different form of a Punnett square by doing what's called a pedigree analysis to track inheritance patterns in families. So I'm going to show you a pedigree of a dominant trait from juvenile glaucoma, so an eye disorder. So let's look at this. So we have a pedigree here, and we have males are squares, females are circles. Anything that is shaded this greenish color is an individual that's affected with juvenile glaucoma. Now, juvenile glaucoma, I'll just make mention, causes uh, optical nerve degeneration, i.e. it starts to break down and you start to lose your sight, eventually leading to complete blindness. And so you can notice what we have here. We have an individual that has a male that has the trait. This person has 1, 2, 3 4 offspring with his partner. And you'll notice that we have 2 individuals that are not affected by glaucoma and 2 officials that are affected by glaucoma. Pretty straightforward. I'm so sorry. They have three kids, not 4 kids. That'd be kind of weird when you look at individual 2 and 3 making a baby together. This is not Alabama or anything like that. So 3 offspring, 2 have the disorder, 1 does not. Now, when child number 3 has a baby with person number 2 here, they produce an effective male because the female here is sending out a dominant trait. This person has 2 recessive traits, thus the dominant is always going to be dominant over the recessive. And you can see the same thing here. The affected female has 2 children with the unaffected male, thus producing 2 effective female offspring. And you'll see that in this dominant pedigree, the dominant trait appears in every generation. So it's in generation 1, generation 2, and generation 3. It is a dominant allele. So if you have it, you have the disorder, right? In this case, it's juvenile glaucoma. Now let's look at what a pedigree for albinism or albino organism. Now albinism is it's not a deadly disease or anything like that, but it's a condition by which the skin pigment melanin is not produced. And so those people that have albinism or they're albino, they lack meaningful amounts of melanin. So their skin is very, very pale. They have red, if they have full albinism, they'll have red eyes, completely red eyes. It's kind of alarming to see actually. And they have very, very pale colored hair. That's what an albino individual looks like. Now, albinism is due to a non-functional allele to producing an enzyme called tyrosinase. As you can imagine, it's an enzyme. It breaks down tyrosine. It's important for pigments and stuff like that. And so that's just albinism. Now, males and females are affected equally, just as a note, and most individuals have unaffected parents. So let's look at this recessive pedigree. So we start off with 2 parents in generation 1. One of them is heterozygous for albinism, i.e. it has a recessive disorder. They have kids, 1, 2, 3 kids. All of them do not display the trait, but 1, 2 of which are carriers for the trait. So one of the parents, we don't know which one, has a recessive copy of the albinism allele, and it transmitted it to 1, 2 of their kids. Now, those individuals that are carriers, as you can see, this individual here marries a individual that is a carrier as well, and they produce 4 offspring, which just sounds terrible. I can't even imagine having 1 kid, let alone 4. But you'll notice that they have 1, 2 children that are affected. They both have albinism, but you notice that the parents don't have albinism. They have 1 unaffected kid and 1 kid that carries the disorder. And you'll notice that if you have a carrier, have kid with a carrier, so first cousins basically, kind of a creepy thing, but that's what it is. They produce 3 offspring. 1 has the trait, and the other 2 are unaffected, and they're not carriers. So recessive disorders, you notice that doesn't appear in the first generation, doesn't appear in the second generation, but you do have carriers of the allele throughout, and it only appears in the 3rd generation where you have a carrier producing offspring with another carrier from a different uh, lineage of peoples. So we can use pedigrees to see how traits track over time. So it's kind of a fun thing. I like looking at pedigrees. I think they're kind of fun. I know these past couple slides are awesome for me because I like talking about pedigrees. I like talking about our Punnett squares. So, all right. So we've been talking about monohybrid crosses thus far. One gene, one trait that's controlled by one gene, and that gene has multiple alleles. But we can do what's called a dihybrid cross, where we're looking at two separate traits controlled by two separate genes that potentially have multiple alleles. Fun stuff, right? So getting slightly more complex, but not really at all. We're looking at, we were looking at, purple flowers or white flowers. Now we're looking at purple flowers, white flowers, green peas, and yellow peas. That's all this is. It's nothing insane, nothing complex, but it does add another level of complexity to your Punnett squares. So let's get into this. So we'll produce true breeding lines for two traits. So we can pretend we have, a trait for eye color, RR, and a trait for, say, height, Y. So we have dominant and dominant crossing, making an individual that's recessive. So now, when you do this cross, so anytime you see an X in between like two alleles, so if this was like big P times big P little p, that'd just be a cross. So anytime you see X between like alleles like this, it just means you're producing offspring. That's all. So in this cross, RR, YY times little r, little y, the F1 generation of a dihybrid cross will always be big R, little r, big Y, little y. So it'll be completely heterozygous for both traits. And they will only show the dominant phenotype. So because again, we're only talking about right now the basics, which is just dominant recessive phenotypes. And what we do from there, we take R, big R, big Y, little y, we self-fertilize and we produce the second generation or the F2 generation. Now, a dihybrid cross of two heterozygotes produces a 9:3 to 3:1 ratio, which is fun. And what does that 9:3 to 3:1 ratio mean? Now, phenotypically, if we're using, say, P shape or P texture, so like round or smooth, and P color, yellow and green, our ratios would be 9 round and yellow, 3 round and green, 3 wrinkled in yellow, and 1 wrinkled in green. That's what we're getting at here. Now, that's the phenotypic breakdown. Now, if we look at what that genotype looks like, we have 9 that are big R, big Y, and something else. It doesn't matter. We have 3 that are big R and something else, either big R or little r, and little y, 3 that are little r, big Y, and something else, either big Y or little y, doesn't matter. And then we have 1 that is little r, little y, little y. So let's conceptualize what this looks like, and then we'll look at a Punnett square. So the parent generation, we have big R, big Y, crossing with little r, little y, little y. That's harder to say fast. Meiosis occurs, you produce gametes, and then you have a cross-fertilization between these two. The F1 generation produces 100% of individuals that are big R, big Y, little y. Now, meiosis from there produces gametes that are either big R, big Y, big R, little y, big Y, or little r, little y. Jeez. I forgot how taxing it is to talk about this stuff. But that's the alleles we produce from a heterozygous individual. Now remember, the gametes that we produce from a homozygous individual always have to be big R, little y. Remember, the gametes only get half the genetic information, so they only get 1 R or 1 Y. They don't get both. So, but again, the alleles here of this F1 generation are a pretty big mixture, 4 distinct combinations of alleles. So remember, to make the F2 generation, we cross 2 F1 individuals. Now, because we're crossing 2 F1 individuals, we have 4 possibilities for the arrangement of alleles together. Remember, it can be big R, big Y, big R, little y, and so on and so forth. So when we set up a Punnett square, we set up basically a 20 cell basically a 20 cell square here, right? Where on the top we put F1 plant times F1 plants. So we're crossing 2 F1 plants that are heterozygous for both alleles. And so this is the allele combination that we just spoke about. Big and big, big little, little big, little little. And we have that on the columns and then on the rows. And so we just fill this Punnett square up. So big R is, so this one is big R because we have big R here, big Y, that's filled. But if you think about this bottom right-hand corner, this one only donates a little R and a little Y, and this one can only donate a little Y and a little R as well. Thus, the resulting genotype is little R, big, little Y, little Y. And you just fill out this Punnett square, right? It's the same exact Punnett square that we did. It's just basically 3 times as, or I should say 4 times as big, right? So nothing crazy, right? Nothing crazy at all, but I know it looks intimidating, especially if this is your first time seeing a dihybrid cross, but it really is just as simple as a regular monohybrid cross. But again, how do we get that 9:3 to 3:1 ratio? So you can see, phenotypically, we have 9 yellow and round, 3 round and green, 3 wrinkled and yellow, and 1 wrinkled and green. 9:3 to 3:1 ratio for a dihybrid cross between 2 heterozygotes. Fun stuff. Fun stuff. So this is our dihybrid cross. Again, it's just looking at 2 distinct genes. You could do a trihybrid cross if you wanted. You could do a quad hybrid cross. It just gets bigger and bigger and bigger. But the idea is the same. What alleles what alleles are in your gametes? Line those gamete alleles up on a box and just fill in the boxes. Because remember, when you produce a gamete, it only has 1 copy or 1 allele from each gene. And those alleles can be identical or different, right? So it just really depends. But again, nothing crazy, nothing weird. Now, our dihybrid process brings us to a really important principle. So we talked about the principle of segregation leading to unique gametes, but the important one was this principle of independent assortment. Now, in a dihybrid cross, the alleles of each gene assort independently. What does that mean? That when we look at this big R and this little r, they sort independently into distinct gametes. That's what this is getting at. It's independent. They're not dependent on one another. And remember, this is all due to the independent alignment of different homologous chromosome pairs during metaphase 1. This is what leads to it. Remember, we have 2 homologous chromosomes line up together on that metaphase plate. They line up independently of one another randomly. So this is how we get this in an assortment. So that's fun. Now, a big part about genetics is the probability and the rule of addition. Now, if you take genetics with me in your coming years at Fisher, you'll do a lot more probability stuff, and you'll hate me for it. But I would be remiss if I didn't mention some math. I say you'll hate me for it because most people hate math. I like math. Math's fun. But you'll probably hate me for it because you probably don't like math. Most people don't like math. But probability basically allows us to predict the likelihood of the outcome of random events. That's really it. So we're thinking about principally what we call the rule of addition. Now the rule of addition says that the probability of either of 2 mutually exclusive events occurring is the sum of their individual probabilities. So what the hell does that mean? Well, let's think about this in context of genetics. When we cross big P, little p with big P, little p, the probability of producing a big P, little p offspring is defined as the probability of obtaining big P, little p, 1/4, plus the probability of attaining little p, big P, which is also 1/4. How do you get 1/4? Well, 1, 2, 3, 4, 1 with, right, 1 with 1, 1 with 1, right? That's 1/4. So it's 1/4 plus 1/4, which if you remember how to add your fractions is 1/2. So you can, this is basically an easy way, I guess I should say easier way. It's a more mathematical way to get probabilities without doing a Punnett square. So this idea of, getting, see this big P, little p, if we go back, just want to go back to our Punnett square, just to show you what I mean by that. This is the cross we looked at, right? Right here. Big P, little p, big P, little p. Now the question I asked was, what is the probability of getting little p, big P, and little p, big P? Well, you can see from the Punnett square, it's 50%, but you don't need to do the Punnett square to get that probability. There's 4 alleles, so the probability of each allele is 1/4. So 1/4 plus 1/4 is 1/2. So for those of you that are more mathematically inclined, you don't have to do Punnett squares. You can just actually do the rule of addition to get your probabilities. Just as a note, I like to throw that in there, just to help you out. Now, I also should make mention of the rule of multiplication. Again, more math, probably don't like it, but tough cookies. The rule of multiplication does state the probability of 2 independent events, both occurring, is the product of their individual probabilities. So when we cross big P in little p with big P and little p, the probability of obtaining little p offspring is thus. The probability of attaining little p from the father is 1/2, i.e. there's 2 alleles. To get one of these is half, right? Half of 2 is 1, right? So 1, right? 1 divided by 2 is 50%, yeah. The probability of obtaining little p from the mom is 1/2, i.e. you have a 1 in 2 chance of getting either of these alleles here. So 1/2. Now the probability of little p is just 1/2 times 1/2. And for those of you that remember, you're multiplying your fractions, you multiply numerator with numerator, denominator with denominator, giving you 1/2 times 1/2 gives you 1/4. Math is fun. So that's the probability and the rule of multiplication. So how you can use just straight math without Punnett squares to get ratios. It's whatever you feel the most comfortable with. Some people feel more comfortable with the math. Most people feel more comfortable with the Punnett squares. Oops. You do whatever feels most comfortable for you, but I wanted to just put that out there so everyone was, familiar with it. So that's the end of our math. But genetics really is a probability. So if you take genetics with me, you'll be doing a lot more probabilities at some point. You'll be doing math at some point. Can't get away from math in science and medicine. You just can't. So love it and know it. So in the last couple slides, we're going to talk about a couple specific types of things. So the first thing we're going to talk about is what we call a test cross. And a test cross is simply what we use to determine the genotype of an individual with a dominant phenotype. So what we do is we cross the individual with an unknown genotype, i.e. big P something, with a homozygous recessive little p. The phenotypic ratios among the offspring are different depending on the genotype of the unknown parent. What the heck does that mean? Well, if I have little p, and I have a homozygous dominant individual, the output of these traits would be always 100% little p, big P, right? That's all this guy can give, right? Now, if I crossed it with a heterozygote, right? So I can remember if I have a dominant trait here, right? And it's say I have purple flower, remember, it can be big P, or big P, little p. So I don't know which one this is, but I can figure it out with a test cross. So by crossing my unknown with a homozygous recessive, I can look at the ratios. So if I have a pure breeding dominant, my output should be 100% purple. If I have a heterozygote purple, I should be 50-50 based upon this Punnett square. So this idea of a test cross is going to be really helpful for you in your VGL lab next week, just as a note. So that's a test cross. Now there are multiple more complex things than what we discussed with Mendel, and I want to just talk about a few of them that are going to be important to you. Mendel assumed that each trait is controlled by a single gene. Each gene only has 2 alleles, and that there is a clear dominant recessive relationship between these 2 alleles. I hate to break it to you guys, but most genetics don't work like this. It just doesn't. Most genetic traits are much, much more complex than what we have here. So most genetic traits don't fall into this category. So let's talk about a couple of these different types. So the first is what we call polygenic inheritance. Now, this occurs when multiple genes are involved in controlling the phenotype of a trait. What does that mean? Well, it means I have more than one gene controlling a trait. And so the phenotype, i.e. height or eye color, is the accumulation of contributions by multiple genes. So height is a great example. Eye color is a fantastic example as well. One trait under the control of 2 or more different genes, not alleles, but different distinct genes. Now, these traits show a continuous variation, and we refer to these as quantitative traits. And what does that mean? Well, look at human height, right? Do I have a perfect? I have a picture of this one. I thought I had removed it, but I didn't. But we can look at the number of individuals in a histogram. So you guys made this in your very first lab, looking at your reaction time, and you can look at the distribution of heights, 5 foot all the way up to monster, and you can see the distribution of these traits, right? It's not like green and yellow here, right? There's a spectrum. Right, there's a spectrum of heights. Now there is a most common height at about 5'8". There are people that are short, and there are people that are monstrous. I say that as a 6'1" tall male. Anyone that's taller than me, I consider monstrous, because people that are 6'7" are huge. Like they really are. But you can see there's a continuum here, right? It's not a discrete trait. There's a lot of variability. There are other things besides your genetics that affect your height as well, but a lot of your height is controlled directly by your genes. So height continuously varies over a spectrum because it's controlled by multiple genes. Next up is pleiotropy. Now, pleiotropy refers to an allele that has more than one effect on a phenotype. So pleiotropic effects are kind of cool, and they're really difficult to predict because you have one gene, say it causes one specialized thing, right? It might do a bunch of other different things inside you, right? So when we talked about signaling pathways, right? Sometimes signaling pathways, you have one gene controls multiple different pathways. And that's what's going on here. One gene can do multiple things within inside one organism. Now, one of the best examples I can give you about this is actually, I'm not going to talk about cystic fibrosis, but I actually want to talk about sickle cell anemia. Now, sickle cell anemia is a pretty, it's not a super common trait disease, but it's not uncommon. But sickle cell anemia it's pretty cool. It's pleiotrophic, and it causes your red blood cells to go from circular to shaped like a sickle or sort of shaped like a banana. And there's all sorts of negative side effects to having sickle-shaped blood cells, including lack of transport of oxygen, your blood cells getting stuck, and so on and so forth. But one of the cool things about people that have sickle cell anemia, I guess the only cool thing about people that have sickle cell anemia, because it's a kind of a pain in the ass disease, but those that have sickle cell anemia are actually almost physically immune to malaria. So those that have sickle cell anemia, the defective gene gives them their sickle cells, but also causes a defective protein on their membranes that doesn't let the malaria parasite infect them. So one gene causes sickle cell anemia and an almost complete immunity to malaria. So that's pretty cool. Now, where sickle cell anemia occurs the most is sub-Saharan Africa. Where does malaria occur the most? Sub-Saharan Africa. Cool stuff, right? So cystic fibrosis is another example, and there's lots of examples of pleiotropy, but my favorite is always sickle cell anemia. It's the most I think it's one of the, malaria is one of the most infectious diseases on the planet. So it's a good thing when you beat back malaria for a little bit. Sickle cell anemia definitely does that. So that's multiple genes, that's pleiotropy, but we can also have multiple alleles. So you can have more than 2 alleles for a single gene in a given population. And this can be like, I have 3, I have 4, I have 700. It just depends. And I say there's actually 700 because there are genes out there that have 700 different alleles. It's kind of crazy. Now, the example I'll give you to this is the ABO blood type in humans. It's a single gene that's controlled by 3 alleles. Now, each individual, as we know, can only have 2 alleles because you have 2 copies of, say, chromosome number 1. Yeah, and each chromosome has 1 allele. Thus, you can only have 2 alleles. And so the number of alleles within a given the number of alleles is limited within a given organism, but they're typically speaking, there are more than 2 alleles for the vast majority of traits out there, just as a note. So ABO blood type is a really good example of a multiple allele system. Next up is dominance that's not actually complete. So we've talked about like a purple allele being 100% dominant over a white allele, but we have 2 distinct types of dominance that are not complete. So we call them incomplete and codominance. And so an incomplete dominance is where the heterozygote is an intermediate in phenotype between the 2 homozygotes. What does that look like? Well, if I crossed a red flower with a white flower, not in our peas, but in a different flower, the resulting flower would be a pink flower, because we have a mixing of red and white. And if you mix red and white together, that gives you pink. So that's what incomplete dominance looks like. Now, codominance is a bit different than this. And the best example for this is the AB blood group. So when you look at blood groups, you have the individuals that are AB, right? So they have, you can have A, AB, O, or B, right, for blood groups. Those that are AB express the alleles for both at the exact same time, the allele for the A blood type and the allele for the B blood type. So it's sort of a mixture, right? They're not mixing together like incomplete dominance is, but they're both being expressed at the same time. So it's kind of a distinct thing, right? In this case, the heterozygote is pink because it's a blending of 2 colors. But in this case, you have just they're both expressed, but they're not mixed together. So they're both distinct in the phenotype. And so you can look at what this physically looks like, right? Parent generation, red and white gives you purple. Now, it's important that we make note that the different way we've described this. So we have big C, big C, big C, big C, but we have the allele for R and the allele for R and the allele for white and allele for white. So neither is dominant over the other. So we can't make this little c because this one is not dominant over this one or vice versa. So we denote this slightly differently. We do superscript big R or superscript white. And the resulting one, because again, they're both dominant because they're both being expressed in incomplete dominance. But the expression is mixed, right? We mix purple and white together, giving us, red and white, giving us pink. So I would like to just sort of wrap this up by just looping back to the ABO blood group. And so this system demonstrates both. We have multiple alleles. We have 3 distinct alleles. The gene has been labeled I. So we have A superscript I, A superscript B, and then little I. So that's our 3 allele system. And we have codominance, where I superscript A and I superscript B are dominant to I, little I should say, but they're codominant to one another. So if you have a person that has A blood type, right, you have their genotype is either big A or big A little I. Those that are blood type B, they have big B, and then big B little I. Those that are AB have big AI. That's the codominant. And then we have the pure recessive, i.e. blood type O, which is little I. Now, the phenotype of this is your blood type, but what is that actually physically telling you? Well, the sugars that you have on your outer membrane of your red blood cells. If you're type A, you have galactosamine. If you are type B, you have galactose. If you're type AB, you have galactose and galactosamine. And if you're type O, you have none because you're recessive. And you can look at this final column if you want to know how they can donate and receives. So if you're AB blood type, you're a universal acceptor, but you can only donate to type AB people. So just as I know. Next up, one of the cooler ones is what we call environmental. Oh my goodness, environmental influence. There's a technical term for this. It's just skipping my brain. But where you can look at traits being expressed differently depending on the environmental conditions. So in this case, we have Siamese cats. They're pretty cute cats. The allele that produces the color of their fur, the enzyme that produces can only occur at temperatures below 30 degrees C. So at temperatures below 33 degrees C, we have the activity of an enzyme called tyrosinase, which produces a dark pigment. Temperatures above 33 C do not produce the enzyme tyrosinase, so there is no pigment. So the environment is directly influencing your phenotype. The genotype is the same here and here, but the environment influences it. Yep. So the last thing is going to be epistasis, where the behavior of a gene products can change the ratio expected by independent assortment, or even if the genes are on different chromosomes that do exhibit independent assortment. Well, what does this look like? Well, we could look to the work of a gentleman named R.A. Emerson. He crossed 2 white varieties of corn. F1 was all purple, and the F2, 9 were purple and 7 were white, which was actually not physically expected. So what did he have? He had white corn with white corn. We had a cross-fertilization event producing all purple. And the next generation, the F2 generation, where we crossed 2 whites with one another, 2 purples with one another, the resulting phenotype was not what we expected, right? So when we did this dihybrid cross before, we expected 9 to 3 to 3 to 1. Well, what he got was 9 purple and 7 white. Well, what the heck does that mean? Well, the reality is for grain color of corn, it's controlled by this gene A and this gene B, and there's 2 alleles for both of them, but they operate in a pathway. So you have a precursor that's colorless, enzyme A, which is encoded by gene A, produces some sort of intermediate, which is colorless. However, if enzyme B is present, it produces the purple pigment. So you can sort of see that in this Punnett square, where unless you have a capital B, right, and a capital A, you don't have the production of color. So you need both to produce the color. That's sort of bucking the trend. So we have 2 genes that are in a pathway with one another that are interacting, and you need both dominant versions to create a specific trait. So just fun stuff to think about here. This is a pretty cool sort of genetic system. So that's going to take us to the end of our very long class on genetics. So if you guys have any questions, I will hang around. I'm going to stop recording.